figure we should start with kind of the basics of it. So what's going to end up happening is we will start with some kind of initiation. That would have been nice to include on this. Well, shoot. This image isn't as good as I thought it was. We're going to have some kind of identifying situation because we need to make sure that our RNA is transcripted at the proper point. So somewhere along that DNA, there's going to have to be some kind of marker for our RNA polymerase to then bind and actually be able to process it. So we need some kind of identification or some kind of marker within it. Then we can potentially get our RNA polymerase to bind. In theory then, hopefully we can actually start our transcription, which is going to have some complications on its own. While it's actually running, in theory that should run relatively smoothly. We might predict that to be very, very similar to DNA replication because that's ultimately what's happening. You're just swapping in different nucleotides. The only real difference is that you're looking at uracil versus thiamine. Did I say that right? Thiamine? It is thiamine.
Yeah, that's good.
And then we'll have to do a transcription, and since I'm running out of space and lazy, transcription end as opposed to a termination. Okay? So we're trying to evaluate each of these intermediate stages and understand something about what's happening within those. Remember, we do have prokaryotes and eukaryotes, and so we do have slight differences. In this in this chapter in particular, we're going to look a lot closer at the eukaryotic system, whereas in previous chapters we kind of just focused on prokaryotes. So we'll start with the prokaryotes, and then we'll do some discussion of the eukaryotes. Hopefully we don't get too far into eukaryotes because I haven't pushed too far into it. I would be surprised if we really got that far in beyond what I'm actually ready for, but we'll see what happens. Once the RNA has been transcripted, then we can go through that translation process with proteins and all that fun stuff. And that would then be chapter 12. Okay? So what they're showing here is our polymerase binding and then pumping out the RNA transcript coming off of this. Other things to be aware of. When we're looking at the RNA polymerase, its RNA is single-stranded. So when it goes to synthesize, we have to be careful which DNA strand we're using. Because if we use one strand, we're going to get a different sequence of nucleotides than on the other strand because of the partnering system. Okay? So that means when we look at our individual DNA strands, we're going to have two different types. And they tend to separate these into two different terms, a coding strand and a template strand. And I'm suspicious that I actually deleted that slide. Let me check real quickly here. Crap, I'm pretty sure I deleted that slide. So it's only up at the top. They come up with a variety of different names popping off of those. Coding strand, template strand, sense strand, anti-sense strand, positive strand, negative strand. If you look at the very beginning of chapter 11, they list them all out. And I do believe personally that it's very confusing to kind of keep track of what they're referencing in each of those cases. The template strand is what you're building the RNA off of. Okay, so that's going to be the one that's going to get matched up with your RNA nucleotides. The the coding strand is then what would look like your RNA strand except with RNA or ribonucleic acids as opposed to deoxyribonucleic acids. Okay? So we have to be kind of careful on the phrasing on that, which does also bring in another piece of information. When we look at this, our template strand and our coding strand, do they necessarily have to always be the same one? Is our template strand always going to be the bottom one? No. We could actually start to synthesize our DNA going on the other strand, just the opposite direction. So our DNA has pretty much double the potential sites for storage of information because we've got it on one strand and we also have it running the reverse on the other strand. So depending on what we're trying to synthesize, our initiation, that finding where we want to start transcripting our information across, could match up with either strand. Once it's matched up and we start seeing that synthesis, that's going to be then defining what's our template strand and what's our coding strand. Okay? So just trying to be aware of that as kind of an added wrinkle when we look at these. The initiation process in our prokaryotes, we're looking at RNA polymerase, so that's our big primer that's going to be pushing everything through. And it's divided into two kind of subcategory names. You get the core enzyme, and then you get the holo enzyme. It's just a naming system, so be kind of careful, particularly some of the sapling questions make reference to it. You have to know what's within each of those enzyme systems. Your core enzyme is what's ultimately responsible for the transcription. So that's going to be the list that they've given you here. Okay. What is that nomenclature system referring to? Alpha 2, beta, beta prime? Any guesses? No. That's a good point. You would think alpha and beta we've seen with sugars and which direction our OH is located. They're names of the protein subunits that make up the RNA polymerase. Okay, proteins or potentially RNA pieces. Okay, so what we're looking at is two alpha units come in to make the polymerase, and then a beta and a beta prime. So all of those things have to be together to make up the core enzyme. Okay. The holo enzyme is now looking at everything within it. You'll notice the one big addition is that we have this sigma subunit that's adjusted with this. The core enzyme is what's responsible for the transcription. The holo enzyme is what's responsible for actually starting the transcription. Right? So our big primary difference is this sigma subunit. Okay? And so that's going to be really, really important. That's going to be what actually finds where to start the transcription. So the sigma subunit has a very tight affinity for a very particular sequence of DNA. Okay? So that, depending on what your sigma subunit looks like, you're going to start to target different sections of the DNA. Once it's targeted, it can now bring in the rest of the polymerase enzyme, and now it can say, okay, now that you're here, go ahead and do what you got to do. Okay? Yeah?
So it goes holo enzyme and then core enzyme?
I don't know what you mean by holo versus core.
So you would have to find the starting core, and then the core enzyme comes in and starts transcription?
Yes.
Okay.
Yeah. So the sigma unit really has a tight affinity for the DNA. All right, so that's going to be what brings our polymerase on, says, boom, this is where you need to start working. So it has a fairly strong affinity for that particular DNA sequence. Okay. Issue with that is if it has a tight affinity, is the polymerase ever going to move? No. So once we actually start the transcription, we need to actually ditch the sigma subunit because it's what's holding the polymerase in place. Okay. So if the sigma unit stays there, we don't get any transcription. So there's different actions that happen that allow for the sigma subunit to be released, and then your core enzyme, your polymerase, can then process through and start actually initiate or start your transcription process. Okay? So your sigma subunit is going to zero in on what they refer to as the promoter locus. Okay? So that's just where, hey, this is important. This is where you need to start the synthesis. Okay? Because if it just randomly jumps in at any given spot on the DNA, we may be jumping in the middle of a section of coding information. So it has to have some kind of initiation or start point. The promoter locus is something that helps identify that beginning point. Okay? And that's what your sigma subunit is going to aim for. Okay? Oh, I did have that thing on there. So your DNA strands get divided up into different names. You get template strand or antisense strand. I'm pretty sure I forgot to check this. I think that one's referred to as the positive strand, but I'm almost positive I did not check that, so we're running a 50-50 guess on that. Your textbook for sure has that clear. And then you've got your coding or non-template strand, because it's not template, or the sense strand. So that's ultimately what's giving you your RNA code. Okay, so it would mirror your RNA code. And then what we can do when we look at our DNA strand, which is what they're showing at the bottom, or what I'm showing at the bottom. Your textbook has a slightly different image. I thought it was kind of watered down with a bunch of extra stuff that I just wanted to get rid of. Your textbook lists a whole bunch of different genes. Okay, so your genes is what's coding for the proteins and all of that, and we need to make sure that that gets translated or transcripted perfectly. before we can actually start that, so that's going to be your transcription start, we have to have something that says, hey, polymerase, come in here and sit at this point so you can start the transcription. And that's where they actually identified further upstream of your coding sequence, okay, of particular sections of information that are consistent between the different genes, which means that's probably a signal or a starter for each of those particular sections. Okay. Within prokaryotes, they've identified a couple different boxes. Okay, so in the yellow, you'll notice that the sequence they give there is TAT. And if you look underneath those, you see a sequence of numbers. Those numbers are referring to a conservation of that particular nucleotide within the sequence. Okay? Meaning, regardless of the gene, 79% of the time, we will have a T. 95% of the time, we have an A. Okay? Because that sequence shows up so frequently, it gets its own particular name. That name, in the case of prokaryotes, is the Pribnow box. Okay? Not to be confused with the Tada box, okay, which is what officially happens in your eukaryotes. Why might it be easily confused? What's the sequence in the Pribnow box?
Same word.
Tada. TATA. Okay. So realize they are slightly different things. I will still probably refer to this as Tada. I shouldn't. So if you catch me, watch out for that.
So it's just two over switch?
In the Tada, they're ignoring the last two. Okay. So that Pribnow box is one of our part of our promoter sequence. Okay. A little bit further upstream, we have another sequence of nucleotides that is also relatively highly conserved. If you take a look at the percentages within there, we do have pretty high percentages relative to possible options, which would suggest that if that percentage is so high, that section of the DNA code probably plays some role within the transcription process. Okay? So what we're primarily concerned about is those two sections. What's going to end up happening with our polymerase is we're going to have our sigma subunit come in, and it's going to identify one or both of these regions. Pretty sure it's the Pribnow that it mainly searches for. And then the rest of our polymerase core enzyme can bind in on top. We now have everything set and ready to go. We need to actually get it initiated, and then the exact details on that we don't have, but it will then start that initiation process probably through a phosphorylation. Once that phosphorylation occurs, we can actually initiate and push forward. Okay? So that's what they're trying to show with your next figure. Your hollow, did I get that right? It is hollow enzyme, is everything, yes. Your hollow enzyme comes in, finds that promoter sequence, so we get a really high affinity between the sigma subunit and the promoter. That binds to the DNA. Once that's bound to the DNA, we're going to start to say, okay, now it's time to start working on this. So to go through and process DNA, do we want it all completely folded up? Nope, it needs to be unraveled. So what we've got at this initial stage, once it binds, is what's referred to as the closed complex, because it's closed. What will happen at that point is your RNA polymerase can then open up the DNA strands. To go through and open it up, what is it going to need to have within it? Some kind of helicase associated with it, which is also going to build up stress on either end, so we're also going to potentially need a gyrase to help alleviate that supercoiling process. We now have the open complex because we've split that open. Okay. Now our polymerase can start to match the new RNA nucleotides into the appropriate template strand, and we can start to synthesize out. One of the things that they noted is the very first nucleotide, for some reason, always happens to be a purine. Okay? Remember, your purines are the larger of our two nucleotides, so it's the double rings. We're looking at which two nucleotides? What are the nucleotides that are the purines? Adenine and guanine. Yeah, I remembered the adenine. I couldn't remember the guanine actually.
I only remember G because GAC is the start code on it.
Interesting. We'll talk about that in chapter 12. It can then put on a few more nucleotides before the sigma unit dissociates away from the complex, and our polymerase can then continue to zip down our DNA strand, continuing to add in our nucleotides. Okay? So relatively not that bad of an initiation process. Questions about that? For it to function, what needs to be present? It's the easiest thing that needs to be present.
DNA.
Okay, fair enough. The DNA does need to be present. The RNA polymerase. How about the ribonucleotides? We're going to have those ribonucleotides at a very high concentration so that it can actually process. If we don't have those ribonucleotides present, it can't actually process because there's nothing to actually put in. Okay? So when we're looking at the chain elongation, we did just talk about this. We're going to have our topoisomerases, which is our gyrase issue, both before and after, relieving the supercoiling issue with popping that structure open. As it runs, we do have this open complex, and that open complex is constantly moving. So instead of referring to a moving open complex, they refer to it as the transcription bubble, because it would have a bubble within the DNA shifting through it. Okay? We do have a couple potential mechanisms of what's happening with that RNA polymerase, or the RNA that's coming off of that. It is getting matched to the DNA strand. So as the RNA polymerase goes through, we do have the option for our RNA to stay attached to the DNA. Okay, that is a potential issue. As it gets longer, it might very well start to get popped off because it has a weaker interaction with the DNA. But then it could potentially wind around the DNA. Okay, why might we want it to wind around the DNA? What's present in our nucleus or the nucleosome? Stuff that breaks it up. So if our RNA actually wrapped around the DNA as it's being synthesized, we could protect it. Okay, so it's less likely to get destroyed. But what's the issue with that? It's actually protected, so I'd say we'd have less likely mutations.
The rate of transcription.
No, that would actually help with the coiling, actually. Where does the RNA need to go? Outside the nucleus. If it's wrapped around the DNA, we then have to unwrap it from the DNA. Okay, so yeah, that may temporarily stabilize it, but then we've got this fight of getting it back off of the DNA. So if you've ever had to deal with vines, winding and unwinding vines from different... Does anybody have to do that? Okay, never mind. Just me. It's pain. Okay. So instead, what typically happens, or one of our theories, is that it just kind of runs off the side, and then that RNA can fold up or potentially interact with other stabilizing proteins to make sure that it's okay.
Or a girl's blow dryer cord.
Garden hose?
Unwinding the blow dryer cord from what?
No, like the way that their cord gets all like super twisted.
I guess I haven't played with a blow dryer enough to know that one.
You guys can't stand it, like it's messed up.
Any case, so this is kind of our general process and things that we're going to have to watch out. The bubble is roughly 17 nucleotides. Okay, so why might we mention 17 nucleotides? Because everybody is now frantically highlighting and writing that number down. Is it really all that important to know that it's 17 nucleotides for the bubble? Why mention the number at all? It's a scale issue, okay? Just to get an idea of how big that bubble is. Okay? How big is that? That's ridiculously tiny when we consider how many nucleotides actually go into a sequence. We think back to the error within a sequencing of our replication. Our errors were on the order of 10 to the 10th nucleotides. Here, our transcription bubble is only 17 nucleotides large, or small, so it's relatively tiny.
On the grand scheme of this micro.
Yeah, on the grand scheme of things, it's ridiculously small. Actually, before we do that, as we go through and continue to process, we don't want to convert the entire DNA sequence over into RNA. We just want to convert the sequence into what we particularly want for our RNA. So at some point or another, we need to be able to stop this process. Okay. There's one thing that we could already potentially consider that's going to help with that stopping process. Okay. What actually kept the RNA polymerase bound to the DNA?
The RNA polymerase.
The sigma structure. The sigma structure isn't there anymore.
So then
Right. So it's going to be a lot easier to pop the polymerase off, okay, because we've lost that kind of DNA holding ability. Okay, so that's kind of a nice thing, so it won't be as difficult to pop this off. But we're still going to need something to help facilitate that because we still need the polymerase to actually polymerize our RNA. So we're going to need something else to help code for that, and that's where we can run into two potential termination options. One of the more common ones is looking at what they refer to as a stem loop or hairpin, followed by a sequence of uracils. Okay, so what ends up happening is in that stem loop, so we're processing out our, I guess they aren't showing it nicely there, our RNA, so we'd match up and we'd end up with this particular sequence, and what will end up happening is that we'll get this inverted matching, which generates this secondary structure. Okay, so that's that stem loop secondary structure. Because the RNA is completely now free-flowing off of this, there's something about that structure that somehow folds back or slows the RNA polymerase. Okay. So on its own, that doesn't stop it, it just slows it down a little bit. So what we need is something to help really kind of push and get that polymerase off. That's kind of what your uracils are there for, with that string of uracils. Why might that help us out? What are those uracils partnering with on the DNA? Adenine. Okay, we're getting a lot of adenine linkages. Okay, why is that going to be important? If our polymerase has now slowed down, okay, we're now giving it actual time to potentially dissociate from the DNA. The interesting feature about our adenine uracil linkage is that that hydrogen bonding interaction is weaker than the adenine thymine interaction. So once we've slowed down the polymerase enough, and then we string in a whole bunch of these uracil interactions, it starts to go, well, there's no reason for me to be stuck here. Okay, we've minimized all of those hydrogen bonding interactions, and we've slowed it down enough that it pretty much pops off on its own through just kind of a thermal relaxation almost. Okay, and then our DNA can fold back on itself, and we're done. Okay, so it's kind of a interesting, we're just going to slow it down enough, and then it falls off on its own. Okay? And that was one of the issues with our sigma complex. Okay? That's why we talked about it back here. When we go through and do this, if there's no sigma complex, we've lost the affinity for the DNA. The only thing really keeping the polymerase on there is that hydrogen bonding interaction. Okay? And if we start to generate a sequence of weak interactions, that's going to weaken the interaction between our polymerase and the DNA, and it can potentially pop off. Okay, so that hairpin loop slows down that translation or transcription. I keep calling it translation. That transcription enough that when we hit that sequence of very weak hydrogen bonding interactions, our polymerase pops off. If it's no longer attached to the DNA, we can't transcribe anymore, and now our RNA strand that we've transcripted can free float off. What is that RNA strand that we just made? mRNA. Okay, it's our messenger that we can then go through and process later on. Potentially, we could potentially see some other ones in there. The other type of termination is what is referred to, where is it? The factor-dependent termination. And this is one of the things that I find kind of irritating within this chapter is we get a lot of these factor-dependent things, and their nomenclature system for factor-dependent is, they just call it FD for factor-dependent. Right? It's a bit silly. In any case, it's a little bit more complex. So what ends up happening in this process is this protein row slides back over the top of the RNA and kind of starts the processing of the RNA and starts to interpret and see what's going on with it and helps unwind the RNA away from the DNA. Okay? It is a pretty fast mover through this process, but it isn't the greatest. Okay? So our polymerase will move faster than the Rho protein up to our termination point. At the termination point, our polymerase slows down. Okay? The Rho protein can catch up and effectively push the polymerase right off the DNA. Okay? And that's what's happening with our Rho protein. So we're looking at an additional protein that comes along and says, get the heck off the DNA.
So those two always go hand in hand? Or
What do you mean by those two?
Well, the protein that stops, termination protein in the Rho, they're always there together.
We will see them together. We need something to slow the RNA polymerase before the Rho can catch up. Okay? Which is kind of interesting because you've got a just standard termination with just the coding, and then we have a type or a second type where it's the coding and the secondary removal.
So it kind of just collides and bumps it up.
Yeah, at least as far as we're concerned. We get a slightly different mechanism, as it's mentioned in the lower left. We're going to see a GC-rich region, and that's going to slow it down. That GC-rich region we can see kind of in the termination. Okay? We've got a lot of GC-rich regions within that fold back. It's that secondary structure folding back that somehow slows down the polymerase. So we're seeing a very similar hairpin loop type issue folding back, slowing down the polymerase enough that the Rho protein can come up and actually hit it. Okay? Yes, this is right. Yeah, we'll talk about it in a little bit later. So we've got the basic idea, our initiation, the actual transcription, and then our termination. Okay, so now what we're going to do is go through and look at things that can affect primarily our initiation and our termination. So what are things that could either enhance or slow that process down? The first thing that your book mentions is looking at alternative sigma factors. Okay, sigma, or that sigma complex, is designed to interact with our promoter region for a particular gene. That promoter region will be slightly different for different genes. If it's slightly different, what does that mean? Different sigma complex. Okay? So within prokaryotes, there's a lot of different sigma complexes. So what ends up happening is we've just changed the protein sequence so that it binds slightly differently for different genes. So we can see the the transcription of different genes depending on the concentration of our sigma factors, okay, or what sigma factors are present. We can also, or what we will also look at, something known as enhancers. We'll look at operons, and then we'll look at transcription attenuation. So what might our enhancers do? Enhance what? You're right. What's it going to enhance? It's going to enhance our ability to actually transcribe the DNA, transcribe our DNA. All right, so really original, come up with enhancers as a name.
What do you mean by like enhance?
So what we're going to try and do is facilitate that transcription process, actually get it started going faster.
Oh, okay. So more efficient.
Then we've got operons, which is looking at a much larger sequence of what's happening. We've got genes that code for regulatory enzymes. Those regulatory enzymes can bind into the DNA or bind to other things that can either facilitate the transcription process or slow it down. So those are really fun and confusing to talk about, particularly with the textbook. But we'll look at two primary situations, the lac operon and the trip operon. And then we'll mention other possible things that we won't discuss. And then transcription attenuation. What's happening in that case?
You have two things that happen at the same time. One leads to the other.
Attenuating, we're just slowing it down. Okay, we're causing some kind of change in the rate of, that's what I thought it meant. But I'm a chemist, not an English major, so I'm not positive. I thought attenuation just meant change, potentially slowing. No one disagrees, so we'll just accept that. Alternative sigma factors, we can also look at viruses. That's about all I'm going to say about those. We can look at different sigma factors. There are viruses that incorporate slightly different sigma factors that will change the replication process. Okay, so they will interact with the polymerase and say, you should transcribe this section of the gene. You should transcribe this section of the gene. You should transcribe this section. Okay, so it's just how they interact with the polymerase. Our enhancers, we're going to be looking at a sequence of DNA that is upstream of our promoter. So our core promoter was our Pribnow box in that minus 10 to minus 35 region. What do those numbers mean? I didn't mention that.
The number
They're the nucleotides back from transcription start. Okay, so that Tada region, or our Pribnow box, is typically in that minus 10 to minus 35 region. So that's our core promoter because that needs to be there. And we had very, very high retention of that across the genes. We can take a slight step back and we'll see another sequence a little bit further back. That was, I don't think we gave that a name. Our minus 35 region. Okay, so we're looking in minus 40 to minus 60, so it's just a step further back from the minus 35. And that can also help and contribute. That's going to be potentially how our sigma complex could go through and bind to that genome. So that's our full promoter section. If we look even further back or further upstream, we can run into these things that they've called FIS sites. What is the FIS site? It's where proteins that have been named FIS can then bind to the DNA. Okay? What their role is going to do is to help speed that process along. Okay? So if we have a protein bind into that FIS site range, it's close enough, or potentially close enough, to interact with our core promoter to help the sigma complex bind. Okay? So it can actually speed the sigma complex, or increases the sigma complex's affinity for the DNA. If it becomes with a stronger affinity, what can end up happening? We're going to potentially see an increase in our transcription rate because we're starting it much, much faster. Okay? Under those same kind of, or that same kind of logic, does something binding to that site have to actually increase the affinity? It could decrease it. And if it decreases, what's going to happen? We're going to decrease the ability to actually transcribe that DNA, and we're going to see less transcription. Okay? In that case, we aren't looking at an enhancer. We're looking at a silencer. Okay? Because it's going to silence the transcription of that gene. Okay? So we've got these enhancers and silencers. Okay? And their definitions are all very, very, very similar, unfortunately. Now we're actually moving into operons. Any questions about our enhancers or potentially silencers? Okay. Operons. So in the case of the operon, we're looking at a much larger section. So we're looking at an operator where everything is kind of being controlled from. So that's how our regulatory genes can get changed, or regulatory proteins can come in and bind to that operator and potentially change how our promoter is seen by our sigma complex and the RNA polymerase. Okay, so that's our operator sequence of DNA. We then have our standard promoter, and then we'll have all of the information that's coded after it, which can be our structural genes. So those are what are going to generate the proteins that could potentially go out and do different processes. Okay? So your operon includes all of this information. These particular gene sequences are strongly condition dependent. Okay? So the whole idea behind this operator sequence is that if conditions are right, something will potentially bind to that operator site, or something won't. Okay? Or maybe something will to help facilitate or destroy the reaction, okay? Or destroy the transcription. So we've got a bunch of extra pieces that now can add complexity to our overall interpretation for our transcription. Within our terminology, we've got an inducer or induction. We've got a repressor or repression, depending on exactly what we're referring to. we'll also have co-inducers or co-repressors, and then we have positive control and negative control. Okay, so what are all these things going to piece together to mean? What is an inducer going to do? Okay, it's going to allow for our transcription. What is it? A repressor going to do?
Prevent transcription.
Okay, prevent our transcription. Our co-inducer or repressor? Okay. That could facilitate or stop our inducer or repressor. So what we're looking at in those cases could be trying to take it back to what we were looking at with our proteins, and I can't remember the opposite. Remember, we have competitive versus non-competitive versus uncompetitive inhibitors. So we could look at our inhibitors that are going to potentially interact with these to change the structure to either allow them to work or to prevent them from working. What's the opposite of an inhibitor? Was it just an activator?
Allosteric.
Yeah, well, allosteric did both, right? It inhibited that interaction between our co-inducer and a repressor back with these is our allosteric interaction because it's changing the shape of the structure, and that is absolutely happening. Inhibitors, and for lack of a better descriptor, we'll call them activators. Cofactor just seems to make it worse, but you're right. Something to actually cause the reaction to go. Cofactors will be important, but I wanted more of a general term. What's our positive control and negative control? What? Positive control and negative control?
Kind of like homeostasis, positive feedback or negative feedback.
So it's either going to I think it's positive if it allows it to go, negative if it stops it. Okay, so very similar towards our inducer and repressor, but our positive control, negative control is looking at the expression of the gene. Okay, this is, I think, gene expression, and this is gene non-expression. Okay. So now let's kind of look at our lac operon. So what we've got from our textbook is an image of our lac operon, which I think we've got a little bit better. Okay. So it's going to code. So the reason we have to start with this is first understand what its role is supposed to be. The lac operon codes for a sequence of genes that allows for the consumption of lactose when there is no glucose. Okay. So it's a very, very selective. Okay, we need to make sure that we have a lot of lactose, but we don't have glucose. Okay, why? Glucose is probably easier to process. We have more things to go through and process that than our lactose. We might as well stick with the glucose. So if we've got glucose, let's just process the crap out of glucose. As soon as we run out of glucose, we need another carbon source. That other carbon source can be our lactose. Okay, so if there's now lactose present, now the lactose can be processed. Okay, so that's kind of what our lac operon is working on. Within, go ahead.
Lactose intolerance?
I have no clue, but quite possibly.
I don't think we can actually use lactose.
Yeah.
Well,
So like lactose intolerance.
This is mostly
means that you don't have. And this right now is for sure in prokaryotes, so it doesn't actually apply to us anyway.
Some actually,
for that matter.
like E. coli actually can produce, break down.
Yeah, it
lactose.
breaks down lactose.
Like in your stomach, they're
Yeah.
actually evolved with this.
Oh,
That's why you
go
get all
back here.
bloated when you drink milk.
Yeah, because you
That's
can't.
one of the reasons.
Okay. Side the point. Don't get me distracted. This is hard enough as it is.
Biology.
So, what we're looking at within our lac operon is we actually have this initial sequence out here. Oh, no, I'm advancing it. Don't advance. Stop it. We've got this initial sequence out here known as our lac-I. What that's going to code for is our regulatory gene. Okay? The regulatory gene does not have to be right next to our main operon. It can be somewhere else. In the case of the lac operon, it is right next to it. Okay? So what will end up happening is we can code for that regulatory gene, and it's going to generate a repressor. Okay? That repressor ends up forming a tetramer. Okay? So we generate a protein that then goes into a quaternary structure to generate our repressor. That repressor can then bind to the DNA at the operator sequence. As soon as it's bound to the DNA, it's actually going to prevent the binding of our RNA polymerase, or our sigma structure rather, and that then shuts down the ability to process any of those other genes. Okay? And so the rest of those genes are involved in the processing of lactose. Okay? So in the case of our inhibitor, what we're doing is just setting up the inhibition saying we don't want to spend energy making a bunch of proteins to process lactose if it's not there. Okay? So that's kind of the goal of this. So we don't actually want those genes expressed. But what happens if lactose is present?
You need to make something to shut that off.
We need to get something through there to actually start processing that lactose. Okay? So in that case, what ends up happening is we'll have an inducer come in and bind to our repressor molecule. When it binds, notice that dramatic change in shape, squiggly versus square, changes the shape. That change in shape prevents it from binding to the operator sequence. Well, if it doesn't bind to the operator sequence, now it's actually exposed, and our sigma structure can come in, have that strong affinity, and we can actually put on our RNA polymerase, and now our genes get coded. Okay? Once they get transcripted, the RNA can then get translated, we've got our proteins, and our proteins can now push through the processing of lactose. Okay? I don't think it's that big of a deal, but just in case, each of those protein sequences is doing something slightly different, but they're all part of being able to process lactose. Okay? The galactosidase, I think, happens to be the primary one. That's our primary processor of our lactose. The permease section of it is what's allowing lactose to actually pass through so that it can get through and actually be processed. And then the transacetylase is also going to be part of activating the lactose so that we can actually process it. Okay? So in the presence of lactose, we can actually form all of those and it's all good to run. Okay? So what kind of system did we do here? An induced mechanism or repression? This is induced because what we've done is as soon as our inducer came in, we actually coded for the sequencing. Okay? Is it positive or negative? This one is positive because, again, we've started the transcription process. Okay? There is another figure in here that will help make sure that we actually got that right. Okay? So that covers the first part of our idea behind this lac operon. Okay? If we got lactose, we want to be able to process it. But remember, the lac operon, we only want to process that lactose if there's no glucose present. Okay? So we need another type of control on the processing of these genes. Because if there's glucose present, we don't want to spend the effort to actually process the lactose. Okay? So we need something else to actually control this enzyme, or control that gene transcription. Okay? Our inducer in this case, okay, lactose is a nice answer. It turns out it's not exactly lactose. If lactose gets high enough in concentration, it gets converted into, I think it's allolactose. Allolactose is what's actually binding to our repressor. Okay? Which I think becomes more obnoxious when we move into glucose. Okay? So that's kind of our positive induction, right? Is that what we decided that one was? Yeah. Okay. Remember, we've got that when glucose is not present. So we're also going to have some other secondary feature that has to contribute to this. Okay? This is where I think it got exceedingly confusing, because then they start talking about the next part of this process. It's still the lac operon. We're still concerned about how it's being controlled. However, you'll notice, does it look the same? No, they changed how it looks. It's still referring to that same thing, but we now have to refer to a slightly different section of that genome. Okay? So in our previous case, we were looking at binding to the operator. In this next case, we're looking at another binding site. Okay? And they're going to refer to that as the cat binding site, and I forget what it actually represents for. So we're going to have a repressor that's going to bind to our genome, okay? And in that case, it's going to signal that we're lacking lactose. Okay, that's what we saw in that last case. Our cat binding site is going to tell us that we're lacking glucose. Okay? It's referred to as catabolite repression. Again, I don't know why they decided to change the names up. Still, again, looking at our lac system. Okay, and if you look, whoa, that's not the button I wanted. If you look really carefully at the phrasing, they call this Z. And if we look way back, we did have a lac Z. Okay, so that is what they're referring to. So what's happening in this case? Our glucose concentration needs to actually be an extra signal within this. Okay, so first off, the basics. What we're going to do is have this cap enzyme, okay, or sorry, a cap protein. In the presence of some signaler, it's going to generate our active complex. That active complex can then bind to our genome. Once it's bound to the genome, it's actually going to facilitate RNA binding. Okay, so let's walk back through what's going on with that. What is CAMP? C-A-M-P.
Cyclic AMP.
It's cyclic AMP. What is that coming from? Okay, what is its role there?
Energy source.
No.
Oh, wait. Oh, yeah.
It's acting as a promoter in this case, okay, to facilitate the translation, transcription, which ultimately goes to translation. Okay. What is that facilitator trying to signal? Remember, in this part of the process, what we're trying to do is get a glucose dependence. So what do you think the relationship is between our cyclic AMP and glucose? There's a high concentration of glucose. Do we want to activate our genome?
Yeah.
No. Remember, with our lac operon, we only want to process lactose when there's no glucose. So if we have a high concentration of glucose, we don't want this process to occur because this is going to speed that transcription. Okay? The relationship between our CAMP and glucose is inversely related. The lower the glucose, the higher the CAMP. The higher the CAMP, it can then bind to our cap protein to activate it. Now it can bind to our DNA, activate or facilitate the binding of our RNA polymerase. Okay, so if everything works in concert, we have a high concentration of lactose. Okay, we can now allow for the processing of those genes and we can start to process it. But if we also have a high concentration of glucose, there's no activation of that gene. If we have a low concentration of glucose and a high concentration of lactose, the high concentration of lactose allows for just the standard transcription of the genome. But the low concentration of glucose facilitates speeding up that process because we drop in a promoter at the same time. So in one case, we're looking at a repressor of our genome that we then stop. And in the other case, we're looking at an activator of our genome that we can start. Okay? Both of them are being involved in the same lac operon, both trying to control that lactose processing within our metabolism. Okay? So if we bust out our options here, your textbook has a fairly nice image, in my opinion, that I had to go back over and over and over and over to try and make sure I understood this. What we're looking at is in our lactose operon, so our standard lactose situation, okay, just looking at our lactose control. Okay, our DNA will translate for a repressor enzyme. That repressor enzyme shuts down the transcription of your genes. Okay, So since it's shutting it down, we're looking at a negative control. If we have the presence of our inducer, we've got an induction of the gene process because the inducer shuts down our repressor, and we can now transcribe our DNA. Okay? So in the standard lac operon description that we started with, it is a negative control because it prevents the transcription of our DNA initially, and it's an induction because we can then induce the transcription of our DNA because our inducer can come in and bind to our repressor. Kind of makes sense? Yeah. It'll take a little bit of processing to kind of get through and try and tie those pieces together. Okay, I'm not going to lie, this took me pretty much all week to try and even sort of make sense to me. Okay? So that's our standard lactose kind of control. We can then add a layer of difficulty on top of that, and there's a second regulatory gene, okay, which binds or creates an inducer. Okay? In the case of that inducer, it is inactive when it's first synthesized. So it doesn't do anything. Okay? When we bring in our co-inducer, in the case of our catabolite repression, our CAMP, okay, the CAMP binds to our inactive inducer, activating it, so it can then bind to our DNA, and we see the transcription process. Okay? So it's a positive control because we're actually facilitating our main gene, our main protein, is causing the transcription. In our other case, it's negative control because our regulatory protein prevents transcription. Okay? Both of them are classified under inducers because our protein needs some kind of inducer to shut it down or activate it to allow for the transcription to occur. Kind of, sort of? A little bit made better? Is it cloudier or clearer than mud? Okay, we'll just accept that. It's tar. It's even worse. What's that? The co-inducer on the positive control is CAMP. Yeah. The co-inducer in the negative control is allolactose. So it's if we have a lot of lactose, we're going to make allolactose. So it's saying lactose is present. We need to get rid of it. Process the genome that allows me to get through that lactose, to eat it. Okay, so that's what's happening with our standard trip operon. In the other case, what they refer to as our catabolite repression, what we're looking at is a lack of glucose. So in the case of less glucose, we want our gene to be transcribed even faster because we now have no carbon source. There's no glucose, so I can't get energy from that. I need to get energy from somewhere, so gosh darn it, give me something. Okay? So now it's saying process the crap out of that genome so that we can actually start to get energy from the lactose. Okay? So what I personally went through and did is went back to just this image and played around with changing our concentrations to say where we ended up going with each of these cases until it actually kind of made sense. Okay? By all means, if you would like to, feel free to analyze that image or that image to see if it makes any sense to you. I don't think it will. Okay? What you really want to do is spend your time looking at this image, making sure you understand what each of those things are doing and how they're facilitating that gene expression. You can then go back and look at those other ones and say, okay, this is what this is trying to say. Okay? Actually, before I do that, you'll notice that we have negative control, positive control, and then just induction. Is there something else we could throw in here? We could look at repression. Okay? In both of these cases, we were looking at inducing the genome when we had all of our stuff. We could also look at shutting down transcription. Okay? And that shutting down process, that's where we can move to the trip operon. Okay? In the case of our trip operon, we've got all these polypeptides and we've got this big fancy image. Ultimately, all it's trying to do is go through and actually synthesize tryptophan. Okay? So when is it going to be important to activate the enzyme or activate the DNA, our genome? I shouldn't say enzyme. When should we actually activate it and have it function? When we don't have tryptophan. Okay? If we have tryptophan, there's no reason to actually run this. Okay? So what we're going to actually look at is our negative, sorry, I need to go back to this, our negative repression. So what we're going to look at as our, it's going to be called a co-repressor at this point because we're looking at repression, being tryptophan or some form of tryptophan. That tryptophan will bind to our repressor, which will shut down our processing. Okay, so what we're seeing is this case. Okay, repression and officially negative induction. We generate our repressor, but that repressor is inactive until we've activated. So we want to process our protein that will then control our genome.
What was it you wrote right there? I'm sorry. The way you crossed out it.
Gene. So we now have that floating around and ready to go. Okay? If we've got a lot of tryptophan, our tryptophan can then bind to it and say, stop producing those proteins because we've got enough tryptophan. There's no reason for it to be there. Okay? At that point, it shuts down. We don't have that transcription anymore. And what we're looking at is a repression of the gene. Okay? And it's a negative feedback because we're actually stopping it once it becomes activated. So our negative and our positive are looking at when it's activated, what happens when we activate our regulatory gene, our regulatory protein, what happens to transcription? Does it move or does it not? Okay. So our trip operon, in my opinion, is a lot simpler because all we're really doing is turning it off. Okay. Questions? Yes, there's questions.
No.
Just playing.
Yeah.
Other things that we can go through and look at is secondary structures. Okay. Those secondary structures can go through and change the speed at which we process the genome. Okay. The speed at which our RNA polymerase moves through. Okay. So we've got a couple different sequences that can come out of it. So it's the secondary structure. They've given a couple different names to these. You get your pause structure, which is looking at what they refer to as a 1-2 loop. So a loop between your 1 and 2 region. They've got a terminator loop. So if we take a look at our 3-4, we've got that sequence of GC-rich range. Okay, what's it? Flip-flopped GC-rich range, followed by a sequence of U's. Okay. And then we also have an anti-terminator structure, meaning, do you see a terminator structure? No. So the anti-terminator just means it can't stop. It'll continue to process. Okay. So what ends up happening within each of these has to do with the speed at which the polymerase moves through the enzyme. Okay. And what they're trying to refer to back with our trip is that right up here in this sequence, we've got a lot of trip codons. Okay, so that sequence of codes are UGG codes for tryptophan. Okay, if we've got a lot of tryptophan, what's going to end up happening is that section slows down. Or sorry, speeds up, because we want to be able to get through it. Okay, if it speeds up, it continues to move through the rest of this relatively quickly. Wait, where are we? Make sure I get this right. There's our stop. Shoot, this one was a difficult one too. Let's try and step back from it. Our pause structure, so what's going to end up happening is if we hit this pause structure, I've got it now. Our polymerase goes through and processes. In prokaryotes, we also typically have the translation process happening right behind it. Okay? If our translation process catches up to our transcription process, what's going to happen? It's going to fall off because you're now butting up against each other and it can't control anymore. So what's going to end up happening is as our RNA is getting transcribed, it's also getting translated. If the translation process hits here and we don't have a lot of tryptophan, okay, that means we can't process this anymore, so it pauses. Okay? And that's what's going to kind of slow down the reaction, which gives this section of RNA enough time to actually form that secondary structure. If it forms that secondary structure, what happens? It falls off. So our RNA polymerase falls off the DNA, and we've now actually stopped the actual synthesis. Okay? What happens if instead we have lots of tryptophan? Okay? It continues to process. Okay? So in that case, what will end up happening is it can actually speed up and catch up, and it can actually prevent our reaction from occurring, or it allows that transcription to continue through. It doesn't actually stop the transcription. So what's happening is that we're seeing this RNA being pulled off of our DNA sequence, and there will be another protein coded after it. Okay? If we want that protein to also be coded for, we'll have an internal mechanism. That internal mechanism can prevent our translator from stopping our transcription. Sorta? So all that we're really saying here is that our RNA can actually code for a pause, and that pause can be enough to actually stop the translation or stop the transcription. Okay? Or if there is no need to pause, it continues through and we don't actually stop, and we can now continue to process through and potentially get the next protein in the sequence. Sort of? So if we go back to our genome, if we're processing straight through this and our polymerase pauses, okay, our translator pauses, it can't catch up with our polymerase, the polymerase continues to process through, okay, and we can get all of those other genes coded for or not coded for as well. Okay. If we don't pause, it continues to process through and we get everything. So we get an anti-terminator in that case, and it can process everything. Okay. Well, that was kind of confusing, but I think that's the best I'm going to get out of it. Okay. At that point, what we now have is the end of our, almost end of our prokaryotes. What we're looking at is an attenuation. And so this is what they're trying to describe in a slightly different image. Okay. We've got our ribosome that is translating our mRNA, and we've got our RNA polymerase on there. Okay. If our RNA polymerase catches up, or our ribosome catches up, it can then stop the process, or we end up forming that stop loop, and our RNA polymerase stops translating, stops transcripting, and our ribosome codes just for the genes that have been spit out. Okay. If it doesn't pause, okay, then it can actually continue to go through and translate. Transcribe. And we can, a longer section of mRNA, the ribosome will continue to go through and process, and we get more genes coming out of it. Okay. So that's referring to the attenuation, the secondary structure of the forming RNA strand can actually slow the processing, and if that process slows, we can then change the end results of the genes being expressed. Yay, eukaryotes. What's awesome about eukaryotes? They're more complex, which makes it even worse. Okay. There are three primary polymerases when we're looking at eukaryotes. We've got RNA polymerase 1, 3, and 2. 1 is primarily there to synthesize your rRNAs. 3 is there for your tRNAs and other kind of small RNA molecules. So it's kind of a processing enzyme. And then your RNA polymerase 2 is primarily for your mRNA. Okay, so that's the one that's going to get translated over into proteins. The other two polymerases are just processing RNAs that will actually act as their own enzymes or do other gene regulation type things. Okay? So no translation coming from those. They're composed of a variety of different subunits. The reason I put polymerase 2 as the last one on this sequence is because we got a handy-dandy table, and that handy-dandy table is in reference to polymerase 2. Why polymerase 2? It's the one we know the most about. Okay? So if we take a look at what happens with our naming system of the subunits within polymerase 2, I found this very entertaining. RPB1, RPB2, what are those in reference to? RNA polymerase 2. We're going to end up with RNA or RPA and RPC. Okay, so they came up with another nomenclature system, and instead of calling them 1, 2, and 3, they can refer to them as A, B, and C as well. So in this case, we're looking at RNA polymerase 2, then 1, 2, all the way up to 12. What's the 1, 2, all the way up to 12? It's a different protein unit that makes that particular name up. Okay, so RPB1 is different from RPB2, different, so on and so forth. Okay, so they were at least consistent, same name with 1, 2 through 12. Okay, each of those are in varying sizes, and each of them have slightly different functions. Some of those functions we understand better than others. You'll notice that we also have an extra little column on the far right saying E. coli homolog. What is that in reference to? Would have been nice if they actually changed the E. coli homolog to it's the mirror for our prokaryotes. So RPB1 is effectively, what is that, beta prime? I know it's not very easy to see. RPB2 is our beta subunit within our prokaryotes. So they're very similar in structure and function. And then we also get something very similar to a sigma structure. That said, we don't officially have sigma complexes or sigma structures in eukaryotes. It's one of our big differences between those. Last thing that I want to tag on here, just because I think it's actually interesting and it might be particularly relevant for your homework, we can see another way to distinguish these. Their response to a poison from a particular type of mushroom. Polymerase 1 does not get inhibited by the mushroom, so it will continue to process. Polymerase 2, it is inhibited at low concentrations, meaning the poison is very, very effective at ceasing polymerase 2 activity. At very, very high concentrations, polymerase 3 is shut down. Okay? So that enzyme, or that poison, is effectively shutting down protein formation because it's shutting down which polymerase? Polymerase 2, and that's the one that's primarily going to be responsible for our translation into proteins. Okay? So that will become particularly relevant. So how does our polymerase actually bind to the correct site? Okay, so this is going back to what we talked about initially with our prokaryotes. What are the different signals within the DNA that allow our enzyme or our polymerase to zero in on it and say, okay, it's time to actually start transcribing this section of DNA. Okay? This is where you get the TATA box. Shows up at roughly minus 25, so about 25 nucleotides upstream from our initiation site. Okay? Why do we refer to this one as the TATA box and the other one as the PRIBNOW box? What's that? Could go back to the other AT. We're looking at a different range in the DNA as well. Okay? The PRIBNOW box shows up in the minus 10 range. Our TATA box shows up at minus 25. Okay? So it is showing up in a slightly different section of the DNA. Okay? Other things that are going to fall within this, we also are going to have an upstream element, which is going to be effectively our enhancer that can facilitate the polymerase actually binding and propagating forward with our transcription. Okay? That upstream element may contain a variety of different options. So that's our enhancers or silencers that we kind of talked about with our prokaryotes. We will typically see a GC box. So we're seeing a lot of guanine and cytosines within that. And then we might also see what's referred to as a cat box. There's a lot of mites on that, and that's because it's not consistent across species or even across genes. They just happen to see those, and that they are present in certain cases, and they are conserved in those certain cases. The INR is our initiator sequence. It is not conserved at all, but it does have a lot of pyrimidines. Okay. And then downstream regulators, you can see those contribute. Why might those not contribute as much? What's downstream of our initiator?
Genes.
The gene that you're actually trying to transcribe. Okay? So we do see that show up in eukaryotes. It's just fairly rare because that's where our gene is. So for it to fold back on itself and then allow the polymerase to go through, it's going to be kind of a complex structural arrangement. Okay? Sure, we might as well end on this slide. So we're looking at our initiation of transcription. So what we'll see is transcription factors, which you'll see them labeled as TFs. They are not part of your RNA polymerase, but they are needed for the transcription. Okay? Kind of actually impressive when we put it all together. We're looking at 2.5 million Daltons as far as the total size for that whole initiation process. Okay? In those other cases, we're looking at maybe hundreds, maybe thousands of Daltons.
What's a Dalton equal to again?
Gram per mole. So it's a pretty massive agglomeration of proteins. Go figure, we have so little understanding of it because it is awfully massive. The initiation will begin when we form that pre-initiation complex. So that's looking at your polymerase, and then 6 different transcription factors will all come together to kind of contribute to that. Okay? Those 6 transcription factors get named, see, actually I'm not positive I remember this. Transcription factor 2, meaning for polymerase 2, and then D being the 4th one. Okay, let's see, did I do that right? We have the next image. Hallelujah, there we are. Okay, so remember the polymerase 2 is the primary one that we'll study. So we're looking at a transcription factor for polymerase 2, and it just happens to be D as the name given to it. Okay, that one binds to the Tata box. And then we'll see a few other different proteins come in and interact with each of those things. Okay? So that transcription factor 2 is made up of 2 separate subunits, your Tata binding protein, and then your transcription binding protein associated factors. Okay? So you'll get all sorts of other factors coming off of that. Okay? So we'll pick up with our eukaryotes on Tuesday. Yay, eukaryotes.